Hey, today we're going to be looking at bacterial genetics and biotechnology. When we start talking about bacterial genetics and bacterial genomes, we're going to be comparing them to eukaryotic genomes, like our own genome. Following that, we're going to be looking at tools used in biotechnology and how they were derived from microorganisms like bacteria themselves. And we'll look into the central dogma, how we go from DNA to RNA to proteins themselves. We'll look at gene expression regulation through the central dogma. Okay, looking at bacterial genomes themselves, here we see a cell on the left. This is transmission electron microscopy. This is actually a pseudo-colored photo, and what we see is the entire genome of a cell that has been displaced from the cell. So that is all the DNA that's packed in the nucleoid, that general region where DNA is found, not the nucleus, but the nucleoid in bacterial cells themselves. DNA stands for deoxyribonucleic acid, and we can see the structure of DNA to our right. DNA is made of four bases: adenine, thymine, guanine, and cytosine. Here we see adenine and thymine bond together with two hydrogen bonds. We see this at the top. And at the bottom, we see guanine and cytosine bond together by three hydrogen bonds, making a slightly stronger bond as there's three of them. DNA takes on this helical structure, which looks quite different from this real image, this transmission electron microscopy image of DNA. But what we're seeing is that this DNA helix is wound up around itself. So if we were to zoom in further on this image, we would be able to see, kind of the winding of the DNA here. Okay. Looking at genomes of bacterial cells, prokaryotic cells, if we're looking at them versus eukaryotic cells, there's a big difference in actually how we structure the DNA sequence of what is the coding region. So in bacterial regions, the coding region, meaning what we take from DNA to RNA to protein through the central dogma, expression of genes, is all going to be in one region. So that's going to be a coding region, a bacterial gene. On the other hand, eukaryotic organisms like ourselves actually separate out these coding regions and have spacers in between them called non-coding regions. So our coding regions are called exons. Our non-coding regions are called introns. Kind of funny because our introns are excised out and our exons are left in. So exons are coding areas, but they're interrupted by our introns. In the process of altering DNA, going from DNA to RNA, we actually end up removing those introns and only having the exons. So the coding region is going to be all together. So it would look similar at that point to the bacterial genome, where the coding region is not interrupted. Okay, something unique to bacterial cells is that they can have what we call plasmids in them. So besides having their own genome, they can have these circular rings of DNA that can replicate completely on their own within the cell. So that means they have an open reading frame and they're able to multiply these plasmids. You can have one plasmid that goes to two plasmids within the cell itself. Plasmids can hold quite a few bases, but they're not as large as the genome. Some of the interesting genes that they can confer for is genes for antibiotic resistance. So what we see a lot of times on these plasmids is genes that are beneficial for the microorganism's survival. And these genes can actually be passed through what we call horizontal gene transfer, meaning from one microorganism to the next, they can transfer this DNA. So it's not only inherited from parent to child in vertical transmission or parent to offspring. When we go from one cell to two cells in binary fission, we're actually looking at transferring these plasmids from cell to cell. So we get this genetic variation with these plasmids. Looking at gene expression itself, there's a couple things we want to remember. So going from DNA to RNA to protein requires a lot of energy. And while protein is the most ubiquitous macromolecule within the cell, cells still have to be careful in how they have to be a little bit frivolous with the energy that they're using. So, when we're looking at DNA, some DNA only goes into RNA and doesn't make its way into protein. It's not necessary. It has functional RNA. Um, and some proteins that we're making are only required at specific times. So we can upregulate the expression of genes and then downregulate the expression of genes. So let's say there is all of a sudden a drought and there's a lack of water for the cell. It can upregulate in a stress response. Stress-associated genes with desiccation or drying out. So it prevents from drying out. So we can funnel the energy to upregulate certain genes or downregulate unnecessary genes so that the cell doesn't waste energy making unnecessary proteins. So looking at how cells respond to their environment, well, microorganisms that are unicellular respond very, very quickly. Part of this is due to the nature of the cell itself. It's very simple, it's very small. Our DNA is not in a nucleus, it's in a nucleoid. And our DNA to RNA doesn't have to leave the nucleus, it just goes DNA, RNA, protein. In generally around the same place of the small cell. So it's very, very fast the way that it can respond to its environment. That's a unique adaptation of microorganisms. So let's talk about gene expression here. We can have induction or upregulation of genes or repression and downregulation of genes. Upregulation means we need more of this gene. So the gene is the DNA version of a sequence. When we go to RNA following that, and then we go to protein. So that's what we're seeing in this picture. Here we see a gene, then we go into mRNA, and then we go into protein. This image is actually showing us the interplay between induction and repression. So here we see we've induced the expression of a protein, DNA, RNA, protein. And then we see over here, this protein is going to interact with the regulatory sequence, and it's going to start repression of a target sequence. So maybe we don't have expression of this gene, or we downregulate the expression of this gene. Let's go ahead and take a look at a common example. This is the lac operon in E. coli. This is one of the most well understood systems in how we can upregulate and downregulate genes. So here we're looking at transcriptional control. Transcription is going from DNA to RNA. So this is the genes itself that are in the lac operon. We have gene lac Z, Y, and A. Lac Z, Y, and A are going to be necessary for the breakdown of lactose. So E. coli can break down lactose if it wants to, but it can also break down glucose and prefers to break down glucose. But if glucose runs out in the environment, it's going to want the ability to be able to break down lactose, or else the cell could die if it didn't have the ability to break down lactose. But the ability to break down lactose and expression of all of these lac genes is not necessary if we're not breaking down lactose. So in an abundance of glucose, we don't want to express these genes because we'd be wasting energy expressing those three genes. So when only in the situation when we're running out of glucose and we have an abundance of lactose, the cell is going to switch over and it's going to upregulate expression of these genes. And actually, the way that it does that is when lactose comes into the cell, we have a little bit of beta-galactose. This is actually a little bit leaky. We have a teeny bit of beta-galactose around that breaks up lactose. And what it ends up doing is making a molecule that's going to interact with this process. And this molecule is called allolactose. So a little bit of presence of lactose being broken down by beta-galactose being separated gives us allolactose, and allolactose interferes with the repressor that's sitting on the gene. So repressor meaning it's stopping expression. So lactose gene is normally not turned on because we typically have glucose in the system and we don't want to waste energy making these lactose genes. So we have a repressor sitting on it. And that's what we see in here, this LACI repressor in red. But when we switch over to using lactose and beta-galactose breaks it down and makes allolactose, Allolactose interferes with the repressor. So we see interference, physical interference, removing the repressor from the gene. So this is a very physical thing. We can look at the stoichiometry here that a RNA polymerase needs to get in here to make RNA. But if we have this inducer sitting on the gene, it's not able to physically interact with the gene. So now that allolactose has interacted with the lac repressor, it's removed it from the gene. The gene is now free for the RNA polymerase to come in, read the DNA sequence, and make the mRNA that will eventually make protein. So this is how we have expression. In the presence of lactose here, when we are not using glucose, the allolactose, which is made from lactose, binds to this repressor. The repressor can no longer bind to the gene, and we have transcription of these lac operon genes. So the operon is just the multiple genes that we find together. Now, when there's no lactose present, we don't want to be making these genes because these genes are going to break down lactose. So if we don't have lactose in solution, we'd be wasting energy by making lac Z, lac Y, and lac A. So this repressor will stay on the gene and it will prevent the gene from having an RNA polymerase coming in and making mRNA. So it prevents expression, it blocks transcription. So again, just really quickly to summarize this, and there's plenty of great videos on this as well. The lac operon is really well studied in how it's going to be constantly repressed and then expressed under the right conditions. So E. coli typically uses glucose. When it runs out of glucose and has lactose present, beta-galactose, which is a little bit in the solution, will go ahead and break down lactose and make allolactose. Allolactose is the inducer here as it interferes with the lac repressor, and that's the lac repressor that we would see here. So again, in the situation of not using lactose, we would be blocking transcription. A little bit of lactose is in, no glucose is available. We want to make more of these genes. Allolactose prevents the binding of the lac repressor, which allows the transcription with an RNA polymerase to make lac Z, Y, and A, the genes responsible for breaking down lactose. So now that lactose is available, we want these genes, we want to upregulate expression on them. Okay. Looking at DNA, DNA is copied from cell to cell and passed down in vertical transmission, and of course in horizontal transmission as well for microorganisms like bacteria. But every time a cell divides, it can sometimes read the bases incorrectly. So here we see DNA placing down the new bases. Well, as it reads a base, so it's as we're attaching our A's to T's or G's to C's, sometimes there's an error that occurs and it places the wrong base down. For E. coli, we have one change for every 10 million bases. That actually adds up to be quite a lot. Sometimes these mutations end up staying, they're fixed within them, and they get passed on to the next generation. So this is how we can start events of speciation and we can start new adaptations of microorganisms. These accumulations of mutations that start changing microorganisms. So mutation is really why we see diversity within organisms. If you change the DNA, you change the proteins because we go from DNA to RNA to protein. Okay. Let's get back to plasmid. Those are circular rings of DNA. Again, these can be transferred from cell to cell in the process of horizontal gene transfer, and in binary fission, they could be passed down. So they can be passed from the parental child to the next generation of the two cells, essentially. This circular ring of DNA can confer genes of interest for survival. Here we see tetracycline and ampicillin. So these can be even resistance to these antibiotics that are passed down. And in purple, we see an origin of replication. That's going to be important because this is how this plasmid replicates itself within the cell. So what's unique about these as well is that they have cut sites on them, so they can be cut up within the cell. And what these cut sites are where restriction enzymes can cut. Restriction enzymes are unique proteins that we find in bacterial cells that cut specific sequences of DNA. So it's sequence specific. Over here, we see all these different types of restriction enzymes. We can see where they came from: E. coli, Haemophilus, Bacillus, um, all of these enzymes here that cut at different DNA sequences. We have some of them that cut straight through, creating blunt ends. So it cuts that phosphate backbone, and those hydrogen bonds can break away once we cut through. Over here, we see them cutting and leaving overhangs. So if you were to separate these, we would have essentially overhangs with one another. Same here, overhangs, and same here, overhangs versus blunt cuts. So these restriction enzymes always recognize these same sequences and always cut there. If we go ahead and look at these sequences, let's read them forward and backwards. Here we see A, G, C, T. On the bottom strand, we see A, G, C, T. These are what we call palindromes. They're read the same way forward as they're read backwards. So these sequence-specific enzymes are cutting at what we call palindromic sites. Same for all of them. Here we see AAGCTT, read backwards, AAGCTT. Because the complementary nature of these bases, it's going to be forward and backwards the same. So these sequences are picked up. These enzymes can scan these areas, lock on, and cut through that phosphate backbone, which is a covalent bond, a strong bond. So you might be wondering, why do we care about enzymes that can cut DNA? Well, most of the things that we have in biotechnology, the advances that we have, come from the concepts of nature. So this is something naturally developed in microorganisms, these enzymes, and we can take these and we can use them to cut DNA. So we can perform genetic modification by editing DNA sequences, by cutting in specific areas. So let's go ahead and look at recombinant DNA. Recombinant DNA is a combination of DNA from different source organisms. So maybe we have DNA from E. coli, DNA from humans mixed together. So we can mix DNA together. So this is part of genetic modification. Let's go ahead and look at our plasmid vector. This is going to be a cleavage site. So this is where our enzyme is going to cut. So here we see we're cutting. We have those overhangs. Here we see AATT, AATT. That's what was bound together. And where we've cut, we know the exact sequence because we know exactly where these enzymes cut. That was that AATT sequence, AATT, that we see right there, that we see again right here. Now, if we want to add in foreign DNA, let's say this is E. coli, and this over here is going to be human DNA that we want to add to our E. coli for protein cloning or something, we can use the same enzyme to cut the DNA of interest. So we see it's going to cut through the DNA, and we can take that sequence with the AATT here, and it's going to perfectly bind. It's going to hybridize with the DNA. So now we have our recombinant DNA. So we'll use DNA ligase, which will glue the molecule together. And now we have our plasmid, a chimera plasmid, which combines DNA from different sources. So here's our original vector. We add it, we cut it, and then we took foreign DNA, we cut it with the same enzyme, we added them together, we ligated them, and now we have our unique recombinant DNA plasmid. So it's a chimera plasmid that we're looking at there. Okay. So this is unique to how we can genetically modify organisms. So one of the reasons why we would want recombinant DNA in a plasmid is that, let's say we took DNA of interest from a human. Let's say we took the insulin gene per se, and let's say we're using a plasmid that's typically growing in E. coli. So we can actually add this circular ring of DNA, this recombinant DNA, back into E. coli. And E. coli can use this DNA to make the proteins from it. Because this DNA codes for proteins, we can know the sequence of what we put in here. So insulin used to be a slaughterhouse byproduct that was gathered from pigs, but the pharmaceutical industry actually picked up on biotechnology and was able to produce insulin from genetic modification like this. So we take human DNA for the gene of insulin, which is better than even swine insulin itself. This is more relevant for human use, more practical, and it's hybridized to a plasmid. So we put this plasmid into cells and we cause these E. coli cells to express that DNA. So we go from DNA to RNA to protein. And that protein is the insulin of interest. Then what we can do is lyse open, break open the cells, and isolate that insulin and purify it. And now we have insulin for the pharmaceutical industry, and we have a plethora of it. So we don't have a limited amount like what was happening when we were using it as a slaughterhouse byproduct. So the pharmaceutical industry uses recombinant DNA and protein cloning like this in order to make quite a few pharmaceutical products. Okay, let's talk about another technique used. It's called polymerase chain reaction. The polymerase chain reaction actually is based off of the natural process of DNA replication, but it's taken into a test tube. So we can look at in vivo DNA replication and in vitro DNA replication. PCR is used for many nucleic acid amplification tests, or what we call assays in the field. This is actually what some of my background is. So I worked at Roche Molecular Systems, which was #1 for assay development. If you've ever taken a PCR test for COVID um, or for other assessments, this is essentially what they're doing. This is the basics of the biochemical tests. So when we talk about DNA replication within a cell, we know that we have our DNA double helix, and it opens up so we can read the core bases on the inside, and we use our DNA polymerase to make more. Well, we're going to do something very similar to that in a test tube, but we're only going to amplify instead of the entire genome, which would happen during cell division. We're only going to amplify a sequence of interest. So if we're looking for a sequence of DNA, maybe from a virus, or if we're looking at a sequence of DNA in a human to see a specific biomarker that they have that may make them more likely to have a cancer, we can amplify or make more of just that specific area. PCR is actually also used in crime scene investigation. So if there's only a little bit of blood splatter at the scene and we need more DNA to work with to be able to sequence the DNA and understand who it could be from, you can run a PCR, polymerase chain reaction. Okay, so let's go ahead and start with our DNA. So here's our 5' and 3'. Our DNA is anti-parallel to one another. We go ahead and we heat it up. When we heat it up, the hydrogen bonds that we find on the inside, those A to T, C to G that we have, they end up breaking from heating it up. So that's what we call denaturation. We denature the DNA molecule. Then we lower the temperature. So we go from 95 degrees Celsius, that's close to boiling, to 55 degrees Celsius. And what happens when we drop the temperature is that our primers that we add into solution bind to our DNA. Primers are complementary. So if this is an A, this is a T. If this is a G, this is a C. So they're going to naturally bind because of that complementary nature of base pair bonding. Then we increase the temperature again to 72 degrees Celsius. And in the solution, we have what we call a Taq polymerase. This is an enzyme that can amplify DNA. So it goes ahead and it sees that primer, it sticks here and knows where to start and starts amplifying the DNA. Now, if you look at this step 3, what we have now is 2 identical double helixes of DNA. What we started with was 1. So we went from 1 to 2 in a very, very quick period of time, in 2 minutes. And what you do after this 2 minutes is you run this cycle again. So you go back to step 1, where you heat up the DNA and it denatures. Step 2, you lower the temperature a little bit, the primers bond. Step 3, Taq polymerase. You increase the temperature, you add Taq pol, and it extends that template DNA. So we went here from 1 to 2 to 4. And as you can imagine, as you keep cycling this 1 to 2 to 4 to 8 to 16 to 32, we are logarithmically amplifying our DNA. And once we get enough DNA, we could use it to work with in a lab, or we could use it for maybe adding to a recombinant DNA reaction. Maybe we need to make more DNA to cut up. So PCR is used in a wide range of techniques, and we can use it to even analyze the sequence of DNA from microorganisms. Something unique about the process of PCR is that one of the biggest challenges was that when we have our sample in a test tube and we heat it up, is that our enzymes don't work at those optimal conditions. Just like we said, microorganisms have their optimal conditions, temperatures, pHs. Well, so do our proteins, even on the molecular level. We have these optimal conditions. So most proteins at 72 degrees Celsius, it's too hot for that Taq polymerase, that DNA polymerase to amplify DNA. So Kary Mullis, who actually invented the process of PCR, had to think out of the box. So what can I do? How can I have an enzyme work in these conditions? And what he actually came up with is thinking about, he thought about looking at archaea. So archaea are prokaryotic microorganisms similar to bacteria, but a lot of them are extremophiles. And he found one that lives in a similar environment to this, temperature-wise, and its Taq polymerase naturally operates under those conditions and doesn't denature, it doesn't fall apart because of the heat. The heat can break apart the bonds. So he was able to pull this Taq polymerase from that organism to isolate it and use it essentially in this experiment. So interesting fact about using, again, what we see in nature as something to use in biotechnology. So here we're taking the process of DNA replication and making it in a test tube, in vivo to in vitro. And not only are we doing that, we're using concepts from microbiology, looking at extremophiles, what unique characteristics do microorganisms have that we could take advantage of for the laboratory. The same thing for these restriction enzymes. These are naturally occurring restriction enzymes. Well, what could we use these unique proteins for? Well, we can use them to cut and genetically modify our sample. And this is how we can get genetically modified organisms, GMOs, like foods or animals. Okay, let's talk about DNA sequencing. So DNA sequencing is done mostly to identify microorganisms. Microorganisms do DNA sequencing mostly, not the entire genome, but on a gene called 16S. So we look at a small portion of the DNA and we read the sequence of the DNA. And because we have variable and conserved regions, we're able to identify microorganisms based off of their sequence. We can also know whether they're related or not based off of the differentiation in their sequence, how different they are from one another. So where they would land on the tree of life. So DNA sequencing here, here we can just see kind of like a simple Sanger sequencing, uses what we call DDNTPs, dideoxy NTPs, and NTPs, nucleoside triphosphates, and we go ahead and we add base by base. So just like we would if we were to be replicating DNA and adding a base and adding a base and adding a base, we do the same thing. So here we see we add a base, A to T. Here's the next one, A to T, C to G, A to T, and we keep elongating the strand. But every time we're adding one, we see that we're stopping here because these are actually fluorescently labeled. So these dideoxy actually terminate the sequence. So we run a PCR-like reaction where we're amplifying the DNA. So here we amplify and it stops at T and fluoresces yellow. Then we go again and we amplify. It adds another T and it gives a yellow fluorescence and stops. We amplify, it gives a green fluorescence and stops. Here we amplify, it gives a yellow fluorescence and stop. Here we amplify, it gives a red fluorescence and stops. So what we end up with is this pattern of this yellow, yellow, green, yellow, red. So it's that yellow, yellow, green, yellow, red that we see right here. So it's chain elongation with these dideoxynucleotides that actually cause chain termination. So it's not just consistent going, going, going. It's going, stopping, going, stopping, going, stopping. And every time it stops, we're able to read the sequence. So we're able to accumulate all this data. And based off of the fluorescent colors, we're able to assess what the sequence is. So here we see the sequence GACTGAACGTGTT. And we can also know the complementary sequence because G binds with C, A binds with T. So that's how DNA sequencing is done. Okay, so we have one question associated with this lecture. Question 1 is, which of the following statements about plasmids is false? So give you a second to read over it before we hit the answers. You can go ahead and pause. Okay, and moving on to the answer for question 1. Which of the following statements about plasmids is false? It says plasmids are essential for survival of the organism. So that is actually false because not all organisms have plasmids. Really, the genome of an organism is all they need. So if we look at the other ones here, these are all what is true then. Plasmids typically contain a small number of genes that are useful only in particular environments. So maybe it's antibiotic resistance. Plasmids may contain virulence genes that confer disease properties on bacterium. So yeah, they can have virulence genes, so they can fight one another with these, or they can fight other cells or mount an infection. And here we see plasmids can be transferred horizontally from one bacterial cell to another. That's correct. Not only are they passed down in vertical transmission, they're also passed in horizontal transmission. So the only false answer here is that plasmids are essential for survival of the organism. They're actually not. The genes that are essential are found on the genome. These are extra genetic material. Okay. Again, here is our study guide to go through and section objectives, and these should be done shortly after the lecture. And that's it.